I'm Dr. Sue Ishaq, Assistant Professor of Animal and Veterinary Studies at the University of Maine. Today I'm talking about microbes on the farm for the University of Maine Cooperative Extension 4-H Oxford County Jamboree.
You might have heard of microorganisms, or microbes as they are nicknamed. These are usually single-celled organisms that are so small you need some sort of microscope to view them. This includes bacteria, some types of fungi, archaea, and protozoa. Viruses aren't technically alive, but we lump them in when we talk about microbes. I have a question for you. Do you think microbes are living in your digestive tract and on your skin right now? They are. Microbes are everywhere, including inside our gut and on our skin. Most of the time, we don't even notice that they are there, even though they are active and helping us in unseen ways. Has anyone seen photos like these, where a human or animal silhouette is filled in with microbes? Have you seen the word microbiome on the news talking about health? Well, I work on microbiomes, and today I'm going to give you an overview of what they are and why they're important. A host microbiome is the collection of microbes that live inside or on another organism host, such as animals, insects, plants, or humans. When we say the word microbiome, we are talking about all of the microbes present, but also all of their genetic material, because microbes have really plastic DNA. They can swap pieces of it, rearrange some parts of their DNA code, or even pick up pieces of DNA they find lying around and use it. Most of my work is focused on the microbes in the digestive tract, particularly in animals, and we can think about our organs as ecosystems for these microbes. Let's start by learning about the anatomy of the gut. To give you a starting frame of reference, humans are monogastrics, meaning we have one stomach. That stomach mostly relies on acid to start breaking our food down, as well as some specific enzymes. Monogastrics tend to have short intestines. Because we have short intestines and a hostile stomach, our gut is not very friendly to microbes. Some animals are monogastric that are more welcoming to microbes. Pigs are good examples of omnivores, and rabbits and horses are good examples of herbivores that are more welcoming. They also have a single stomach, but their intestines are longer, and they have this extra pouch, which is circled here in purple, called the cecum. It acts as a fermentation chamber or compost bin and houses the microbes which are really good at digesting fiber in your diet. The digestive tract in poultry is more complicated in order to break down seeds they eat. The crop is a small muscular pouch which stores food and allows the enzymes in saliva to start breaking down the diet. The proventriculus mixes the food and adds acid, much like our stomach does to break food down. The gizzard grinds food and makes sure to get any nuts and seeds broken open. Chickens also have a cecum for microbes. The top-of-the-line digestive tracts belong to ruminant herbivores, like cows, sheep, goats, or deer. They have 4 chambers in their stomach, and the largest one is called the rumen. It's a huge microbial paradise where a lot of digestion takes place. There are also modified ruminants, which only have 3 chambers to their stomach, like alpacas or camels. Here's another question. Microbes in the digestive tract are the only reason animals can fully digest which foods? Check any of the answers that apply. Grass, hay, grain, apples, or silage. All of those foods require microbes in the gut to help digest all or some of the nutrients. That's because all of these foods are plant products and they all contain plant fibers. Plants are made from some very complicated chemicals, including various types of fibers that they use to give themselves the physical structure they need to grow upright. You might have heard about how important fiber is in your diet, or told you need to eat vegetables, and that's because plant fiber is a really healthy thing to eat. But you actually can't digest plant fiber by yourself. No mammals can digest plant fiber. We don't have the enzymes to break fibers apart into individual nutrients. We rely on certain microbes in our gut to do it for us. And it's a good thing they do because they produce some very valuable byproducts that we can use, like energy, proteins, or vitamins. So there is a lot of benefit to having a digestive tract that is friendly to microbes that can help you digest your diet. The rumen is a microbial paradise. It is kept at a nice warm temperature, it is a nice neutral pH, and it is mixed regularly so microbes can access more food or moisture. Importantly, the rumen will keep churning food until it is broken into small enough particles to pass into the next chamber. In order for your cells or the animal cells to access the nutrients in the diet, they need to be broken down into their individual pieces, sort of like taking a puzzle apart so you can rebuild something new by putting the pieces back together in a different order. Rumination can really help with this. Food is regurgitated and it gives the cow or sheep another chance to chew up plants into smaller pieces and add more saliva, full of digestive enzymes. Rumination also gives microbes another chance to break down these plant fibers into individual sugars. Since ruminants are the most complex digestive tracts on the farm, let's walk through the stomach compartments a little more. The rumen has a lot of papillae, which are these points along the inside of the rumen wall. They provide extra surface area so the animal can absorb as many nutrients across the rumen wall as possible. They also make great points for microbes to attach and live on. In a study that I was part of, we found that bacteria are present in the rumen of calves even on the first day of life. The bar chart here shows populations of bacteria at the phylum level of classification, which is very broad. Each color shows a different bacterial phylum. That community changes over the first few weeks of life, and it is somewhat different if you look at the bacteria in the middle of the rumen compared to those living attached to the rumen wall. The reticulum is the second chamber in the rumen stomach and is very similar to the rumen. It has a honeycomb shape to the wall and uses special muscle contractions to help shape food into balls. These are regurgitated so the cow can ruminate and chew cud again. When particles are small enough, they are able to sneak through the reticulum to the next chamber. Even though the reticulum has similar conditions to the rumen, it holds a slightly different bacterial community because of differences in how easy it is for microbes to attach to the wall and how quickly food moves in and out. The omasum chamber is for water reclamation, since eating grass and hay is thirsty work. The omasum has many folds like pages in a book, and when it squeezes, it can reabsorb water and electrolytes to help make more saliva. The bacterial community in the omasum does look similar to the other chambers, but food doesn't stay in here for long, so it is likely that it is just picking up microbes that wash through from the rumen and the reticulum. You can see some of those small slivers of color disappear. These are more rare bacteria, which require more friendly conditions that they don't find in the omasum. The abomasum is the most similar chamber to our stomach because it adds acid and digestive enzymes to the mix to break down any proteins, fats, or carbohydrates into smaller and smaller pieces. It's a very hostile place, and only a few very specialized microbes can survive living here for very long. If you look at the entire digestive tract, you can better see the pattern of the gut as ecosystems. Each column represents the bacterial community found in calves at a different location in their gut and on different days. All the way on the right, we start in the rumen, and as we move left, we go through the digestive tract and out as feces. You can see that the color profile of those bacterial communities change as we start in the rumen, go through those 4 stomach chambers, and then process through the intestines and out to the feces. You can see the section with the most colors, or the most bacterial diversity, is in the middle of the small intestines, or the proximal jejunum. This is where the nutrients in the diet finally become the most available, and it supports many different kinds of microbes. Now we know that there are microbes living in the gut of animals, but how do they get there? Anyone that has spent time around baby animals knows they try to lick everything. That can be one way to pick up microbes because microbes are everywhere, in food, water, soil, bedding, in the air, and on farm surfaces. Most of the time, animals can pick up microbes from the environment around them without the animal getting sick. Baby animals also acquire microbes through contact with other animals or humans. We all constantly shed our own microbes, and these can be passed to others by touch or through the air. Most of the time, this happens without us or the animal getting sick. Baby animals can also acquire gut microbes through milk, where microbes transfer from the mother to the milk and then to the baby. These microbes provide a head start and are very helpful at digesting that milk diet. Colostrum is the first milk that is produced in the first 2 days of life after birth and is chock full of helpful things. It contains antibodies to help target infectious microbes. It contains those beneficial bacteria to help digest the diet. And it contains compounds that reduce inflammation and help the immune system calm down. This combination of ingredients helps the baby's immune system to learn to tolerate gut microbes so it doesn't constantly try to get rid of them all. As humans and animals age, they are weaned or transitioned from a milk diet to a solid food diet. The different components in that diet will recruit different microbes that particularly like to eat that food. This graph shows bacteria at the family level of taxonomy, found in the rumen stomach of lambs, and each color represents a different bacterial family. The columns on the graph represent groups of lambs, and the black dividing lines are added to show the different diets that each group is eating. The first two columns show the lambs when they are eating milk replacer, which is powdered milk for livestock. The next two columns show them eating hay and grain, and then the last four columns show the bacteria in the rumen when lambs were eating grass in a pasture. We can see that the bacterial community somewhat stays the same in terms of that light blue coloring, the Prevoteliaceae family, but often they pick up and lose bacterial families as they transition from one diet to the next. I have one last question for you. Do you think the same microbes that live in the digestive tract could also be used to break down compost? Some of them can. The digestive tract contains very little oxygen in it, and most of the gut microbes prefer it that way. Oxygen can be toxic to them, so they don't like living out in the open. But they can survive in compost piles in which some bacteria and fungi process and remove the oxygen, and these oxygen-free microbes can then set up shop and continue breaking down organic matter into compost. Farms generate a lot of organic waste material, like feces, bedding, or leftover feed. This material is no longer usable in those forms, but they are a treasure trove of nutrients. In the same way that microbes in your gut can break down diet into individual nutrients, microbes in organic waste can break the material down and eventually create soil. Using microbes, we can complete the cycle and recycle nutrient building blocks right on the farm. Plants grown on the farm can feed animals, and their waste can be used as compost to feed that soil and grow new plants. By using microbes, we can make farms more sustainable and self-reliant. Now I'd like to share a little bit about how I got into science. When I first went to the University of Vermont as an undergraduate, all I wanted to be was a veterinarian. The books I'd read or movies I'd watched made it look like an exciting career, and I couldn't think of anything else I wanted to do. But when I got to college, I realized that I had formed a different idea about being a veterinarian, and that maybe it wasn't actually the right career choice for me. Not knowing many other careers that involve animals, I spent my undergraduate time taking a lot of different classes, from anatomy to conservation to ocean ecology. I had a rough idea that I wanted to do graduate work and do research, but I still wasn't sure what I would study. After looking into different programs and applying to many different schools, I was accepted back at the University of Vermont to do my PhD. While I was there, I focused on the microbes in the digestive tract of the moose and how they could be used to develop a probiotic to help lambs grow. After I finished my degree, I went on to short-term research positions in Montana and Oregon, working with microbes in different ecosystems. As of last year, I'm an assistant professor at the University of Maine, and my lab focuses on gut microbes. I'll be working with livestock, wild animals, mice, and also microbes in culture. Here's one of my lab spaces, and you can take a virtual tour of that space by following the link. This room is set up to research DNA from different microbes. This is my other lab space, which is set up for microbial culturing. I can grow microbes under different conditions to figure out who they are and what they do. You can also take a virtual tour of this space by following that link. In the same way that I had a very different idea about what it was like to be a veterinarian, I learned that my idea of a scientist or researcher was also different from what it's really like. There were things I thought would be important to being a scientist, which turned out to be helpful but not required. In fact, there were many other skills that I found were more helpful than you would think. Here's just a few of those. Finally, no research can be done alone, and I'd like to thank all the people on my science journey who helped with the project shown here today.